Hap1 knockout cells are made using CRISPR. A guide RNA is designed, usually to an early exon. This guide directs Cas9 to cut, making a double-stranded DNA break. The DNA break is repaired by the cell's endogenous repair machinery in a process called non-homologous end joining. This is an error-prone repair mechanism that often results in small insertions and deletions. We isolate clones with frameshift mutations, which usually lead to a premature stop codon. Under normal circumstances, the DNA is transcribed to mRNA and translated to the wild-type protein. If Cas9 makes a cut in the DNA, and this is perfectly repaired by the endogenous repair machinery, we still have wild-type mRNA and protein. If Cas9 makes a cut, and this is repaired causing a silent mutation, then this is a conservative change, so there is no change to the amino acid sequence. The cut could also be repaired with an in-frame change. This causes a change to the amino acid sequence, so we get a novel sequence in the protein. All of these proteins are functional. This protein might be functional. When we make Hap1 knockout cell lines, we check for frameshift mutations to avoid these situations. We confirm the presence of the frameshift mutation at the DNA level and provide you with the sequencing information and primers. Let's look at possible protein fates in Hap1 knockout cells. Here's the wild-type protein for comparison. If the repair causes a frameshift mutation which leads to a premature stop codon, there are several possibilities. One possibility is NMD, nonsense-mediated decay, which leads to no protein. Another possibility is that the stop codon results in a truncated protein. Here, the protein is truncated at the C-terminus. Whether an mRNA is destroyed by NMD or produces a truncated protein might depend on NMD efficiency. If there is a premature stop codon, it is possible for translation to start from an alternative start codon after the premature stop codon. If that happens, we get an N-terminal truncated protein, which skips the parts of the protein before the stop codon. The reading frame is conserved, but one exon is skipped. Splice variants like this are hard to predict and come in many forms. It is difficult to anticipate what happens on the RNA level without some qPCR or RT-PCR, so this example just indicates one possibility. Repair can also cause a frameshift which leads to a stop codon after a novel coding sequence. The green sequence is novel, and we end up with a truncated protein with a novel sequence. If there is no protein, there's no function. The truncated proteins are likely non-functional. Let's look at how an antibody might recognize the protein from each of the possibilities for Hap1 knockouts. Here's the wild-type protein, which is detected by this antibody to the N-terminus. Here, there is no protein because it has been degraded by NMD. When we have a truncated protein, it can be recognized by antibodies to the N-terminus, but it won't appear at the expected weight on a Western blot. If the protein is truncated with a frameshift leading to a different amino acid sequence, then it will be recognized by antibodies to the wild-type portion that remains. You can imagine that, if the novel sequence was longer, the protein would be larger. If the protein approaches the size of the wild-type protein, then Western blot results could be misleading, because the band could appear close to the size of the wild-type protein. If there's a splice variant, like exon skipping, the protein won't be detected if the antibody is against a skipped region. If you're using an antibody to another part of the protein, the Western blot for the same protein might look different. Here's the wild-type protein. If there's no protein, there's nothing to be recognized by the antibody. If there is a truncated protein, or a truncated protein followed by a novel sequence, no protein will be detected. If there's a splice variant like exon skipping, it will be recognized by antibodies outside the variant region. You can imagine that if exon 1 was a bit smaller, then the sizes of the wild-type protein and the splice variant could be very similar. This could lead to misleading Western blot results. It is important to use antibodies which have been validated in your lab with appropriate positive and negative controls. Using non-validated antibodies can give misleading results. For example, this antibody is not specific because it also binds to other proteins in the protein extract, like this one. This protein is in all of the protein extracts and is a similar molecular weight to the wild-type protein of interest. This can lead to misleading bands in the Western blot. Even when the wild-type protein is not present, there is still a band. The The protein that remains may have loss of normal function, and the only way to confirm this is through a functional assay. You should choose a functional assay specific for your protein, assessing a function which is important for your downstream experiments. Please see this reading list for some of the functional assays researchers have used with their hap1 knockout cells. Let's imagine this protein is a protease. We can use a protease activity assay to assess the protease's ability to digest casein. This liberates the amino acid tyrosine. In our assay, Folin's reagent reacts with the free tyrosine to produce a blue chromophore, and we see a color change. The more tyrosine released from casein, the stronger the color. Here are 3 situations in which the protein is not functional. When there's no protein, if the functional domain is missing in a truncated protein, if a truncated protein with a novel sequence doesn't function as a protease. In these cases, the functional assay will not show a change in color. When there is a splice variant, the function will depend on the protein that remains. If exon 1 was crucial to this protein, there will be no protein function. However, if the functional domain of the protein is intact, there might be full or partial protein function. It is very rare to detect protein function in a knockout cell line, but if this happens, please contact technical support. This is just one example of how a functional assay would help you to identify a functional knockout. Whether you're using a hap1 knockout cell line or a knockout cell line you've made yourself, here are some tips. If you can, use a functional assay for the targeted protein. In all assays, use the parental cell line as the control to account for cell line-specific effects. If you use antibodies, make sure they have been validated and consider appropriate controls. If you have any questions, please feel free to reach out to us at technical support by email, phone, or chat.